Don't worry if you've never heard of nucleic acid immunity or P1-PDC. Almost nobody working in cancer immunotherapy has. But I think that understanding these are going to be critical for optimizing cancer immunotherapy. And in this video, I'll explain why, starting with the concept of nucleic acid immunity. Nucleic acid immunity starts with the origin of single-cell organisms billions of years ago. Ever since then, there have been infectious agents like this bacteriophage trying to insert their own genomes to reprogram the cell to hack its genome. Nucleic acid immunity refers to the defenses that evolved in bacteria to protect the integrity of their genome against infection, and then evolved much further in vertebrates into mechanisms that also protect our genomes, and which we can now harness to attack tumors. The bacteria that survived evolution have multiple different nucleic acid immune defenses to rapidly and specifically destroy or inactivate the viral nucleic acids. And these include, for example, nucleases, DNA restriction and modification systems, and CRISPR. Some of those defenses were not maintained during evolution to more complex multicellular organisms like insects and worms, which evolved RNA interference and receptor mechanisms to restrict foreign nucleic acids. These systems in this green box evolved to protect host genomes by directly destroying or cutting or modifying foreign nucleic acids. These mechanisms are incredibly powerful for protecting the host, and also they are the foundation for all of molecular biology and for almost all nucleic acid therapeutics. The nuclease RNase H enables gapmer antisense oligonucleotides. DNA restriction enzymes are essential for molecular biology and our entire biotech industry. We are harnessing the CRISPR-Cas system in humans to treat disease by directing it to targets of our choice. We harness RNAi with synthetic small interfering RNAs that we design to selectively cleave targets to treat different diseases. And we keep discovering exciting new ways to use these amazing tools, such as through RNA editing. But as cool as all of that is, the most highly evolved mechanisms of nucleic acid immunity are several families of nucleic acid immune sensing receptors in this red box. When they sense foreign nucleic acids, these receptors induce secretion of more than a dozen different type 1 interferons, which activate and coordinate other defenses against infection. Perhaps the most incredible of these defenses are the CD8 positive T cells, which uniquely can kill infected cells without harming healthy tissue by detecting a single foreign antigen. For cancer immunotherapy, we want to harness these CD8 positive T cells to kill tumors. We're trying to trick the immune system defenses that evolved to detect the sneakiest retrovirus into thinking that the tumor is actually infected by a virus, so that CD8 positive T cells will be produced that are able to attack and destroy the tumor wherever it is spread in the body. In some patients with cancer, the tumor already has been infiltrated by CD8 positive T cells, and they can be unleashed to kill the tumor by blocking checkpoints such as PD-1, and even better if you also block LAG-3. But most patients don't have enough effective anti-tumor CD8 T cells to benefit from checkpoint inhibitors. One approach to treat these patients is to provide exogenous T cells, such as TIL and CAR T therapies. These approaches have yielded some stunning successes, but they're incredibly toxic and expensive. A much simpler alternative would be to generate the anti-tumor CD8 T cells endogenously in the tumor and the draining lymph node by in situ immunization. Usually this involves an intratumoral injection of a type 1 interferon inducer, but systemic delivery is also being explored. This concept is often referred to as making cold tumors hot, because type 1 interferons have been associated with response to checkpoint inhibitors in inflamed tumors. In situ immunization uses the tumor as a source of neoantigen to induce and stimulate new anti-tumor CD8 positive T cells, without us having to go through all of the work to sequence the tumor's genome and try to figure out what the neoantigens are. It induces the immune system to do all of the work for us, just as it would do in the case of a viral infection. If this approach works, then personalized cancer vaccines become irrelevant. So there's been a lot of interest in this. More than a dozen type 1 interferon inducers have cured cancer in mice. But in human clinical trials, the response rates are usually well below 10%. So something is seriously wrong with this concept. I think that what is wrong is that there are 17 different genes for type 1 interferons in humans, and most people assume that it doesn't matter which one you induce because they all bind the same receptor. But it does matter. They are not interchangeable. The best understood type 1 interferons are interferon alpha, for which we have 13 functional genes, and interferon beta, for which we have 1 gene. These bind the same heterodimeric type 1 interferon receptor, IFN-a, but with very different affinities. Interferon alpha has a relatively low affinity, while beta is about 100 times higher. Now, whether it's because of these differences in affinity or something else, interferon alpha and beta have strikingly different therapeutic effects in humans. In mice, they don't seem to differ as much. Interferon alpha is highly effective in curing hepatitis C virus infection. It is approved to treat some types of cancer, but if it's given to people with a classic T-cell-mediated autoimmune disease, multiple sclerosis, it aggravates the disease. And gain-of-function mutations in the pathways leading to interferon alpha production are known to drive CD8-positive T-cell-mediated systemic autoimmune diseases such as lupus and psoriasis. Now, we don't want to induce these diseases in cancer patients, but we do want to induce a controlled autoimmune attack on the tumor neoantigens. And interferon alpha should support that much better than interferon beta. Beta has little activity against hepatitis C virus or cancer, and it actually ameliorates multiple sclerosis. There's evidence that it enhances IL-10 secreting regulatory T-cell activity, which is most definitely not what we want in fighting cancer. And gain-of-function mutations for beta can cause death, but it's from inflammation, not CD8-positive T-cell-mediated autoimmunity. From these facts, we can deduce that when activating innate immunity for cancer immunotherapy, when we want to make cold tumors hot, we should seek to maximize interferon alpha production relative to interferon beta. In fact, since beta binds the receptor with so much higher affinity, it actually may interfere with interferon alpha. And essentially, every nucleated cell in the body can make interferon beta in response to pretty much any kind of disruption, including infections, tissue damage, or cell stress. It's a pretty low bar for a cell to make IFN beta. In contrast, the production of IFN-alpha is highly restricted to a rare immune cell called the plasmacytoid dendritic cell. PDC only secrete large amounts of IFN-alpha in response to activation through 2 of the nucleic acid immune sensing receptors, TLR7 and TLR9. There are 2 primary ways that TLR7 and 9 distinguish foreign nucleic acids from self. First is their location in the PDC endosomes, where they are sequestered from the cytoplasmic RNA or the nuclear DNA. Viruses enter PDC and other cells through endosomes, so this is a great location for these receptors. And second is their receptor ligands, their structural specificity. TLR7 is activated by U-rich RNA, which will be in viral or retroviral RNA, but it's not activated by pseudouridine, which is used in endogenous tRNA and rRNA, the most prevalent RNAs in the cell. TLR9 is activated by unmethylated CpG-DNA, which is in DNA viruses, but in vertebrate genomes, CpG is suppressed, and the C are generally methylated. So this enables TLR7 and TLR9 to detect viruses or retroviruses, which are a danger to our cell's genome integrity. This is the core concept of nucleic acid immunity. So to maximally induce IFN-alpha for cancer immunotherapy, TLR7 and TLR9 are the two receptors we should focus on. The other receptors are unlikely to be highly effective for cancer immunotherapy on their own. These approaches should still be studied, but in combination with TLR7 and TLR9 activation, because the myeloid and tumor cells expressing these other receptors can promote or inhibit those interferon alpha responses. And so we need to understand those effects to optimize human therapy. So first, how to activate TLR7. TLR7 binds RNA as a homodimer at two sites that are circled in green on the sideways view. The first site on top recognizes a guanosine, and the second site binds U-rich RNA, especially poly-U, but not pseudouridine, present in endogenous tRNA and rRNA. And this fact was key to the design of successful COVID mRNA vaccines, which use modified U's to avoid TLR7 stimulation and IFN-a secretion that would inhibit the protein expression from the vaccine. On the other hand, vaccines to treat cancer probably should induce as high IFN-a as possible, and so these should probably use U instead of pseudouridine. Not all cancer vaccines do this, but unless they induce IFN-a in some other way, they are unlikely to have optimal activity in humans, at least if we accept the conclusions I've reviewed so far. Most of the current clinical development of TLR7 agonists relies on small molecules that are a lot easier to develop as drugs compared to RNA. And Aldara was approved long ago, even before we knew about TLR7, for topical treatment of small, local skin cancers. But Aldara rarely induces systemic regression of other lesions, and that's what we're looking for in cancer immunotherapy. Multiple other small molecules that activate TLR7 with a variety of approaches have also failed to induce systemic tumor regression in the clinic in more than a small percentage of patients. One reason they may have failed is that small molecules only bind TLR7 at the first site. They have a very different biologic effect, inducing much lower production of IFN-a compared to RNA ligands. Based on this receptor biology, we can predict that optimized RNA ligands to stimulate TLR7 should be superior to the small molecules for systemic cancer immunotherapy. Of course, since native free RNA is rapidly degraded by nucleases, delivery is going to be critical. For TLR9 agonists, there are also multiple different structures that have gone into clinical development, and the major ones are CpG-A, CpG-B, and CpG-C, shown here with their very different biologic activities and their different efficacy in cancer immunotherapy. These structures represent classes of molecules, each with many thousands or billions of different DNAs that could be synthesized. I'm just showing examples of some that have been tested in human clinical trials. CpG-A DNAs are unique because they look the most like a virus. They have a native DNA backbone and poly-G regions on both ends that form G quadruplex structures that are also present in the genomes of retroviruses and viruses. Because of these structural features, CpG-A DNA induces the highest interferon alpha production in human cells of any published immune activator. And this G10 CpG-A already has shown efficacy in treating cancer when delivered in a virus-like particle, and I'll come back to that. CpG-B DNAs have proven to be outstanding vaccine adjuvants. Two different examples are in two approved vaccines, including the hepatitis B vaccine Heplisav, which is highly effective and has an excellent safety record in more than 60,000 humans. However, CpG-B DNA rarely worked as a monotherapy to treat advanced cancer, and it failed 2 phase 3 clinical trials in lung cancer in combination with standard of care chemotherapy. CpG-C is probably better than CpG-B for treating cancer, but it also had no meaningful monotherapy activity and failed to show benefit in a phase 3 clinical trial in combination with ipilimumab. So I think of these different TLR9 agonists, the pathway forward for cancer therapy should be the CpG-A type of design. To protect the native DNA backbone against nuclease degradation, the G10 CpG-A has been encapsulated in a virus-like particle that is assembled using the purified coat protein of a bacteriophage, Q-beta, and this forms vidutolimod, a non-infectious VLP. The bacteriophage VLP has no receptor on human cells. Like all VLPs, this structure is highly immunogenic, and so the first injection stimulates antibodies to the Q-beta capsid protein, and on subsequent injections, these antibodies bind the VLP, forming immune complexes in vivo, and these immune complexes then are taken up by any cells that express Fc receptors, which includes most immune cells. But the effects vary greatly depending on whether the cell expresses TLR9 to be activated by the CpG inside the VLP. To the PDC, this structure of a VLP with CpG-A inside is associated with the most dangerous viral or retroviral pathogens that require the strongest CD8-positive T-cell responses to eradicate infected cells. And so the PDC are activated to secrete IFN-a. Now, human myeloid cells don't express TLR9, and so they aren't activated through that pathway. In fact, they actually can suppress the PDC response. In mice, the VLP delivery is more effective than naked CpG for inducing regression of uninjected distant tumors. But since mice express TLR9 in their myeloid cells, those results have to be taken with a grain of salt. Fortunately, the human data also have been positive, both in neoadjuvant melanoma and in the very challenging setting of metastatic melanoma that was resistant to PD-1 blockade. Vidutolimod monotherapy in these patients with refractory advanced melanoma induced tumor regression in 9 out of 40 patients, 22.5%, including systemic responses in the 4 patients who had visceral metastases. The other responding patients didn't have systemic lesions. And 2 of the monotherapy responders had progressed on prior combination therapy with the oncolytic virus TVEC in combination with pembrolizumab, which is consistent with the hypothesis that vidutolimod can trigger a qualitatively different anti-tumor immune response than TVEC. But the monotherapy responses were short-lived, less than 6 months. In contrast, in the arm of this trial where vidutolimod was combined with pembrolizumab, the response rate was similar, a bit higher, but the duration was much longer, over 2 years. This suggests that the vidutolimod is initiating or expanding anti-tumor CD8 T cells, and the PD-1 blockade is extending the response duration. And these responses were systemic. The mean reduction in tumor volume was virtually the same in the uninjected tumors as it was in the injected tumors. This is in marked contrast to TVEC, where responses are usually just local. When we looked at the baseline biopsies of the patients whose tumors did not respond to the combination therapy, we saw higher signatures of macrophages. So again, this is consistent with other data that myeloid cells can suppress PDC secretion of IFN-a and clinical response. In addition, there were no RECIST responses to vidutolimod in combination with PD-L1 blockade with or without radiotherapy when we treated 29 patients with refractory non-small cell lung cancer. So these combinations are not going to help most patients with advanced cancer. And these data lead to the question of whether there might be another way that we could stimulate the PDC to overcome this suppression. One alternative approach is to stimulate the PDC using an optimized TLR7 ligand. These data are from a public data set analyzing tumors using single-cell RNA-seq, where I've queried the data to see which of these tumor immune cell populations express TLR7, TLR8, and TLR9, since these are all nucleic acid immune-sensing receptors that are expressed in endosomes of various immune cells and can easily be stimulated using synthetic nucleic acid ligands. Red circles indicate higher expression of the gene compared to the darker blue, and the size of the circles indicates the percentage of that cell population where the gene can be detected. You can see that TLR9 is pretty much only expressed in the PDC, at least among these cell populations, and this is consistent with lots of other data. TLR7 is much more highly expressed in the PDC than TLR9, and it's also expressed in some of these other myeloid cell populations. If those populations are suppressing the PDC secretion of IFN-a in patients, then a TLR7 ligand could overcome TLR9 resistance. And TLR8 isn't expressed in PDC, but it's in most of the other myeloid cells, so it too is a logical candidate for stimulation to try to overcome myeloid immune suppression. RNA sequences that can differentially activate TLR7 or TLR8 or both are already known. So now I'm going to summarize and then introduce a new complexity. The PDC is the key to IFN-a. And it's a very complicated cell because it can suppress or stimulate immunity. And so we shouldn't be surprised to learn that there are different subsets of PDC, including one called the P1 subset, which is primarily responsible for IFN-a production and presumably drives the CD8 positive T cell response. Conversely, the P3 subset doesn't make IFN-a and instead drives CD4 positive T cells and more of a Th2-like effect. And so considering this, we might deduce as a kind of working hypothesis that patients who have a high ratio of P1 to P3 PDCs in their tumors might have better outcomes. And that's exactly what has been reported in this study of 32 patients with melanoma who were naive to immunotherapy. The solid line on the left shows that patients with higher P1 PDCs in the blood had higher overall survival. And the solid line on the left shows that patients with higher P3 PDCs in their tumors had much earlier tumor recurrence than those with lower P3. The senior author of this paper, Dr. Asport, kindly shared the information that the checkpoint LAG3 is expressed on some of the tumor-associated P1 and P3 PDCs. So this study also provides further support to adding LAG3 blockade to PD1 blockade in human clinical development of these approaches. So, of course, this study needs to be repeated, but these data fit the working hypothesis that to optimize cancer immunotherapy, we should seek to induce the highest possible ratio of P1 to P3 PDCs. And so next, let's look at some data on how to do this, at least in vitro, in normal purified PDCs. This table summarizes data from the study by Al-Kalembre et al. That first described these PDC subsets, where they looked at the percentage of purified PDC that differentiated into the P1 and P3 subsets in response to several different stimuli, including the three main types of TLR9 agonist, CpG-A, B, and C, flu virus, and a very potent small molecule for activating TLR7, R848. GM-CSF is a kind of negative control since it doesn't induce IFN-a secretion and it doesn't stimulate P1 PDC. Agonists for STING, RIG-I-like receptors, and TLR3 were not included. It's already known they don't induce much IFN-a secretion from the PDC. The CpG-A is the strongest P1 inducer. It was the only agent that induced more than half of the PDC to differentiate into the P1 subset, and it gave the highest P1 to P3 ratio of more than 10. So this is consistent with the other data I've shown you. Flu virus is interesting because, like most viruses, it suppresses IFN-a induction. Oncolytic viruses are often designed to avoid IFN-a production because it suppresses viral replication. But this defeats the purpose of using them for cancer immunotherapy. Also, on repeat dosing, antibodies to the oncolytic virus will form immune complexes, and those will be taken up into immune cells through Fc receptors, just like the example of vidutolimod, only in this case the immune complexes will activate different TLRs, at least in the case of RNA viruses like flu. So these issues need to be thought through in developing oncolytic viruses for human therapy. And optimized RNA ligands without all of the hassle of an infectious virus could turn out to be superior. These studies need to be performed since no purified RNA ligand was tested here, and they should be extended from purified PDC to PBMC and especially to tumor-associated immune cells. So finally, what conclusions can we draw from these data in terms of how to optimize innate immune activation for cancer immunotherapy? I believe that we should stop, or at least deprioritize, the development of the primary IFN beta inducers, except in combination with agents that induce P1 PDC. And I put P1 in quotes because we should expect that many more PDC subsets will be identified, especially in the tumor microenvironment, once investigators look using single-cell RNA-seq and various multiplexing tools that are sensitive enough to study these rare cells. Likewise, for studies to modulate tumor-associated macrophages and myeloid-derived suppressor cells, these should be performed in the context of their effects on P1 PDC, especially tumor-associated human cells, to find conditions and combinations that will optimally unleash the PDC ability to stimulate anti-tumor CD8 T cells. TLR7 small molecules have failed repeatedly against advanced cancers in human trials, and I think we know enough now to redirect those efforts to RNA ligands that will bind both the first and second activation sites on TLR7. If in situ vaccination succeeds, then there's no need for personalized mRNA cancer vaccines. But in the meantime, these should be designed to optimize P1 PDC induction. I would like to encourage immuno-oncologists to develop RNA ligands for TLR7 with or without TLR8 stimulation and TLR9 stimulation, and to test these in humans with strong translational studies so that we can learn which patient populations will benefit from different combinations. These studies could start with intratumoral injection, but most patients with advanced cancer don't have easily accessible tumors for injection. And so even better would be the development of systemic delivery methods to target tumor-associated PDC and other immune cells throughout the body. We need to think of oncolytic viruses not only for their ability to infect tumor cells, but also for their effect on the P1 PDC and other immune cell populations through the various immune sensors, especially with repeat dosing, where immune complexes will be formed and taken up by Fc receptor expressing cells. These studies mostly can't be done in rodents. Well, they can be, but the data are not going to predict human efficacy because of the huge differences in receptor expression and function that I mentioned earlier. For example, STING activators work much better than CpG as a monotherapy in mouse models, but in humans with advanced cancer, response rates to STING agonists as a monotherapy are generally below 5%. Dose finding and formulation studies, of course, need to be done in mice. These are the kind of box-checking experiments that I mean. But wherever possible, we need to use human cells, especially fresh cells, since frozen PDC lose viability. Normal human PBMC can be used to screen new combinations for their effects on P1, PDC, and other cells. And then the most promising combinations can be tested in vitro using human tumor-associated cells. For example, biopsies of patient tumors, or for patients with peritoneal carcinomatosis and similar conditions, it's possible to test effects in the patient ascites cells, which contain PDC and some of the other tumor-associated immune cells. I think these proposals will improve our chances for developing better cancer immunotherapies to help more patients. Finally, I'd like to thank some of the friends and colleagues who have helped me think through these ideas, and especially how to explain them. This doesn't mean they endorse the ideas, but I appreciate their time and help. If you're still watching this video, I'm guessing you found this interesting. I hope it's also helpful to you, especially if you work in cancer immunotherapy. And so now I want to ask for a favor. See, I'm fundraising for the Dana-Farber Marathon Challenge team. I'll be running the Boston Marathon on April 17th. And our team of more than 500 runners are hoping to raise more than $6.5 million to support young investigators whose research should help to advance cancer therapy. If you go to rundfmc.org, click donate, and enter Art Krieg, I'd appreciate any contribution at all. And also, if you'd like to reach me with any questions on this video or ideas, please reach out on Twitter or LinkedIn. And if you work in or with biotech, please encourage your company to join me and my 20 biotech colleagues who are raising money in the Timmerman Traverse for Life Science Cares to support our communities in ways that go beyond the drugs we develop to impact issues like homelessness and poverty. Thanks for your help.